Hey, hello everyone. We are going to take a look at how science works, the nature of science. And you have studied this over and over since you were probably in kindergarten, the scientific method. So this will be mostly a review. I'm expecting it to be a review. So if you have any questions, make sure you talk to me. By now, you should have downloaded the document, and you can either type the notes into the document and save a copy for yourself, and you can pause the video as you're typing, or you can print the document and write your notes on it. You can also write on binder paper. Just don't forget at the end of the notes, after the vocabulary, there's some critical thinking questions. So you will answer those after you've gone through this video. All right, let's get started. This is very timely because we are in the middle of a pandemic and we're talking about vaccinations to fight the coronavirus, and they're being tested right now. So I want to take you back to the late 1800s when there was a very scary disease called smallpox. And our first vaccination in the world was for smallpox. And you can see in that picture, it was a terrible disease. So it was actually so bad that 1 in 10 children born in Europe died of smallpox. And those that survived the disease were left with terrible scars. Wherever there was a pustule, they could get a really bad scar. There was no cure for it. And so, what a lot of people did is they would take their children to somebody in their community that had contracted a mild case of smallpox, and they would take some fluid from the pustules. They would put it on their children, hoping that their child would also get a mild case and survive. Parents were terrified of this disease. Now, at that time, there was a British doctor named Edward Jenner, and he made this very interesting observation. He noticed that the ladies who milked the cows, the dairymaids, they could get this cow disease called cowpox, and they would get sick from it, but they would never get smallpox. And he thought there had to be a link between these two diseases. So what he did is he intentionally gave a young boy, it was actually his gardener's son, cowpox. And then after the boy recovered, he then gave the boy smallpox, and the boy never got sick. And you might think that is unethical. How could they take a child and do a scientific experiment on them? But you have to remember the times. And the gardener was willing to hand over his son because his gardener was terrified that his child would get smallpox and possibly die from it. So, different time. And actually, smallpox is one of our success stories. And we didn't continue using cowpox, that was the original vaccination. But eventually, they used a different vaccination that produced much milder symptoms, and smallpox was eradicated from the earth by 1980. There was no smallpox moving around in any country in the world. So it actually does still exist in 2 places. The Center for Disease Control in Atlanta, Georgia has smallpox, and it's under deep freeze. And then the equivalent of the Center for Disease Control in Russia also has it. And because the 2 countries don't trust each other, neither one of them will get rid of the smallpox virus that they have in deep freeze. Unfortunately, it'd be good to just get rid of it. So let's go over some basic rules in science. First of all, science is constantly testing things. It should be different researchers can all run similar tests and come up with similar results. And they evaluate each other's ideas, conclusions. Sometimes they reconsider things. They're constantly discussing and arguing ideas in science. So the arguing, I don't mean like fighting, I mean just discussing. So one thing that you have heard over and over is a hypothesis. It's a testable prediction based on either your prior knowledge or your observations, or it could be from research that you've done. You've learned about what's known about this topic and you make a hypothesis, a testable prediction. And you can either support or reject your hypothesis by completing an experiment. Now, a lot of people get that confused with theory. A theory is based on many, many experiments, high amounts of data. It's widely agreed upon in the scientific community that this is true. So things like the theory of evolution, the theory of plate tectonics, theory of gravity, those are theories. Hypotheses are different. Hypotheses could be rejected. They could be wrong. A theory is so widely tested, nobody's going to find it to be wrong unless some major new information is found. So again, discoveries should be reproducible. They should be, it doesn't matter who tests it, they come up with similar findings. And you've seen charts like this. You start with a question like Edward Jenner did. Why are these dairymaids not getting sick with smallpox? You formulate a hypothesis. I think it's because they're getting cowpox. You test a hypothesis by talking your gardener into handing over his son to you, and you collect the data, you interpret what that means, and you share your findings. So here is Edward Jenner. Again, he made the observation of the dairymaids. That's usually based on your five senses. Oops, what did I do here? And so he's got his observations, and he came up with a hypothesis. So it's based on preliminary explanations. And remember, you can find a hypothesis to be false. So actually, they're more often found false than supported. So here is Edward Jenner's hypothesis. Exposure to cowpox would grant immunity to smallpox. And he had to test that. But let's pause and talk about variables in an experiment. And this is going to take you back to biology and chemistry when you learned about independent and dependent variables. So just a reminder, a dependent variable is the variable being tested. It's changed by scientists. So in Edward Jenner's experiment, it was cowpox. He's going to purposely give cowpox to a child. The dependent variable you're measuring because you think it's going to be impacted by the independent variable. So he is going to test if the child gets smallpox, if he stays healthy. Now, all other possible differences need to be held constant. So if you're testing 10 people, everything should be the same about those 10 people. Same age, same health, same amount of exercise and sleep and diet. Of course, that's impossible to do. So we will talk about that later. How do we overcome the difficulty of controlling all the variables? So in an ideal experiment, you will have one variable, and that is the independent variable. So that's how we run controlled experiments. So we test a hypothesis under controlled conditions. We're testing a single variable, the independent variable. Everything else is exactly the same. The experimental group gets the independent variable and a control group does not. So you would have children that you give cowpox to and children that you do not give cowpox to. And then you expose them all to smallpox and see what happens. Sounds terrible, doesn't it? And Edward Jenner did not do that. He did not have a control group in his experiment. But he did have his gardener's 8-year-old son who came down with cowpox, recovered, and then didn't get smallpox. Now, I told you that science should be reproducible. So he gave the child, the boy, 20 inoculations of smallpox, and not once did the boy get sick. Sounds like it should be good enough, right? He reproduced the experiment. So he did come up with the conclusion cowpox gives immunity to smallpox. But he went to communicate. So scientists are very good at communicating, or they should be good at communicating. It's very important. You publish your results, other people read it, they look for errors and biases, and you will argue over it, like your view compared to other people's views. You share your ideas. And they did not like his experiment. They said he needed more proof because he only tested one child. That is not enough in science. What if that child, I don't know, maybe his mother had smallpox when she was pregnant with him, and that's why he has immunity to smallpox. Maybe he had a really light case and nobody noticed it earlier, and he's already immune to smallpox. It wasn't the cowpox. One test is not enough. So, because it is impossible to keep an experiment down to one variable, to control all the variables except the independent variable, there's always going to be errors. That's where we use probability. So, probability is the likelihood that a result occurs simply due to random chance. So, because people are not all the same, or any other organism you're testing something on, they're not all the same. You cannot control all the variables. That's why we increase the sample size. Edward Jenner couldn't run this test on just one person. That wasn't enough. He had to go find more people to do it. When they're testing the new coronavirus vaccinations, they can't run it on just a few people or a few hundred people. They are testing this on tens of thousands of people. Every vaccine is being tested on tens of thousands of people, large sample size. So, Edward Jenner very easily found more parents willing to volunteer their children, and he also tested it on his 11-month-old son, gave them all cowpox, then exposed them to smallpox, and none of them got smallpox. So finally, he published his findings, and this is all very interesting. Why did he call it a vaccination? Because the Latin word for cow is vacca, and we still use the term vaccination years later. Now, not all experiments can be controlled experiments where you have a control group and an experimental group, an independent variable in the experimental group, no independent variable in the control group. That's just not always possible. Sometimes it might be more of a natural experiment where you go out and observe, like Jane Goodall observing chimpanzees in Africa. You go in and you just observe what's happening, you take notes, and you try to come to some conclusions. The advantage of this is it's much more realistic. If you put chimpanzees in a lab and observe them, they are not going to behave in their normal way. But often it's hard to figure out what's leading to their behavior. You're sitting there observing in their natural environment, but what's causing them to behave the way they do? It's hard to know. And bias is always a huge problem. We want to limit bias. So, bias is an unintentional preference that an experimenter has. So, we conduct blind experiments. In a blind experiment, the subjects don't know, if they are in the control group or the experimental group. So right now with the testing of the coronavirus vaccines, Kaiser, I know, is asking for volunteers. They're part of this study. You don't know. Are you going to be given the real vaccination or one of them that's being tested? Or are you going to be in a control group where they give you a needle with a liquid in it, but it actually doesn't have any of the vaccination in it? So that's a blind experiment where you don't know because there's something called the placebo effect. If you think you are getting medicine, you will feel better. So, and it's amazing the effect. Actually, even if you know you're taking a tablet with no medicine in it, you will feel better. It's amazing. The power of the mind over the body. Could your immune system be slightly stronger because you think you got a vaccination? It could. So that's a blind experiment. Now, double blind is when the experimenter doesn't know either. So the subjects don't know, are they getting the real vaccination or just some fluid? And neither does the experimenter. The people giving you the vaccination have no idea which one you're getting. There's just a number on a vial, and that's what they stick in your arm. So, very important to eliminate bias in science. All right, we are done. Your job now is to answer the critical thinking questions and be ready because one of the FRQs that you will take on the AP test in May is going to ask you to design an experiment. So, one of the free response questions is always design an experiment. And you will see those throughout the year. I will have you practice by writing out, designing an experiment as part of an FRQ. All right, I will see you in class tomorrow.